So my job this morning is, that's my understanding, at least, to introduce you a little bit more into single molecule localization, super resolution microscopy. And I want to do a mix of giving you a little bit of an overview of the foundations, but then also talk about some of the applications we have recently developed, or some of the advances we have made on the technology development side. So just a brief outline here is what I plan to cover in the next 45 minutes or so. So after talking briefly about the concept and just saying, having a slide or so about data, the different probes we are using and the data processing we are using in single molecule localization microscopy, I want to focus a little bit on 3D imaging, what the challenges are and what the solutions are for it, and then go into a kind of a case study of the 4Pi SMS microscope from our lab, and give you a very new cell biological example of how we apply this to investigate the organization of the Golgi apparatus. And then in the remainder of the talk, I want to focus more on multicolor imaging. And in that context, also want to go a little bit deeper into DNA paint imaging, and then into a flash paint imaging, which is a development from my lab from last year, which is a multiplexed super resolution microscopy advancement. So when we talk about single molecule localization microscopy, we need to obviously localize single molecules, and this is a very old concept, right? So that we are using this in particle tracking for decades already, and what we do here is we have an isolated molecule for which we want to determine the center of the diffraction limited image, an image like this one here, with the idea that if we find its center, we know the position of that molecule. And we do this by fitting a model function typically to our intensity distribution of that single molecule we have imaged. So that now gives us the position of a single molecule, but that's not a super resolution image, right? That's just the position of a single particle that we can, for example, use in particle tracking. So how do we now get a super resolution image out of that? And let me illustrate this here at this simple kind of simulation or example here. Let's say we want to image nuclear pore complexes and have labeled here nuclear porins here in the nuclear pore complexes on the nuclear envelope. If you image this with a diffraction limited microscope, so each nuclear pore complex has 32 copies here of this labeled nuclear pore complex protein. If you image them all at the same time, they blur into each other. And the reason is that the nuclear pore complex is about 100 nanometers in size, while the diffraction limited resolution is about 250 nanometers, right? So we don't have the resolution to resolve that. So if we zoom into a single nuclear pore complex, the same is true, right? So we cannot distinguish the individual, we cannot resolve that ring. If, however, we have turned off 31 of the 32 locations here, or fluorescent markers here, we will still see a diffraction limited spot here. But now we know it's a single molecule, and we just need to find its center position, and we just plot the center of that molecule. And then we turn off this molecule, activate the next one, image that one, and repeatedly do this until we have localized a large number, ideally all, of these labeled positions here. So the resolution we get with these microscopes is about roughly 10-fold better than what we get with diffraction limited microscopy. So 250 nanometers becomes 25 nanometers, 700 nanometers in the depth direction becomes about 700 nanometers becomes about 70 nanometers. We often talk about the localization precision, and that can be a little bit confusing. So what we actually get from localizing molecules is the localization precision, and that is about 2.5 times better than what we normally would consider being the resolution. I mean, we can discuss about that later in detail if you're interested in, but if you read papers or see claims, it's worthwhile to wonder look into what they are talking about. Do they talk about localization precision or resolution here? So what does this localization precision depend on? Well, it primarily depends on the signal to noise ratio. And this is a formula you see quite often here. So the localization precision is roughly equal to the size of the Poinsett function divided by the square root of the number of photons. If you go a little bit more in depth here, and this is not a very nice formula, but I think a reasonably accurate one by Bernd Rieger and Joerg Stalinga. So you see that localization precision depends on the size of the points, the diffraction limit of Poinsett function, and the number of photons, but it also depends on the background noise per pixel here and the pixel size. The pixel size, and that's very important to keep in mind, the pixel size has a small effect on the localization precision. We normally consider this negligible, and the pixel size is not limiting your resolution. You can have a 100 nanometer pixel size and still get 10 nanometer resolution. Okay, so that's just from a conceptual perspective here for the students in the audience. So these are the terms that you need to keep roughly in mind when you do localization microscopy. So the field, the super resolution version of single molecule localization started in 2006 with 3 papers coming out in parallel. There was Sam Hesse's paper on FPalm, Eric Betzik and Harald Hesse's paper on Palm, and then from Xiaowei Zhuang's lab, there was the Storm paper, and we have, I think, Mark Bates here at the conference, who was actually one of the authors of that initial Storm paper here. And so if you want to hear more about how this all started, I encourage you to talk to him. In the following years, there were quite a number of variants and versions of this. Since there were already 3 acronyms, everyone decided to give it their own additional acronym on top of it. We ended up with quite a lot. What now crystallizes nearly 20 years after these initial papers is, I think, that most people use these 2 acronyms, PALM or D-Storm, D-Storm, to when they refer to these different methods here. So what probes can we use for single molecule localization microscopy? There is the photo-switchable or photo-activatable fluorescent proteins, and in that context, in particular, MEOS 3.2 and MAPLE3, I think, have been used quite extensively. But most people, I would argue, nowadays are using organic dyes. We have photo-activatable or blinking dyes, for example, spontaneously blinking dyes that Luke Levis mentioned yesterday. And I think Luke also contributed here to these photoactivatable Geneva fluors over the last years. So that's one option. The initial Storm paper used these activator reporter pairs. They linked an activator, for example, Alexa 405, with a reporter dye, the Psi5 here, and could thereby achieve the blinking of the Psi5 dye. It's important to keep in mind that for these probes, they have the disadvantage that in the beginning, they're all in an active state. So you first need to kind of switch them off before you actually can see blinking of just a small minority of these dyes. And you usually require some oxygen scavenging buffers to really get the best blinking properties out of these dyes. A couple of years later, Mike Heilemann and colleagues figured out that the Storm probes, or the Storm concept, also works without that activator dye. You just need a slightly higher laser power for that activation laser. And so you can just use Alexa 647 or Psi5 or some of these dyes here. You still need these buffers, and you still need to switch them off, but you can just now use essentially regular secondary antibodies with these dyes. And then the pain probes came up. Actually, it already started also in 2006, but I think the real breakthrough as a generally usable tool, I think, came with the DNA paint development. I'll talk more about DNA paint towards the end of my talk. What's important to note is that all these rely on some photo switching or photo conversion concept where essentially light is turning molecules on and off, while the pain probes rely on a transient binding effect to get the switching effect. So it's a very different switching concept. So it's also interesting to consider which of these are lifestyle compatible. I would say there are probably exceptions here to these rules, but overall I would say it's primarily these top ones which are lifestyle compatible. Most of the probes are essentially limited to fixed cell imaging with some exceptions, and most of the applications we see in the single molecule localization microscopy field are in fixed cells. So how does this blinking now look like, and how can I actually tell good from bad blinking data here? And this is actually very important. We'll come back to that a couple of times later if you want to get a high quality single molecule localization data set. So here on the left, you see high quality data set. On the right, you see a poor quality data set. And that actually, if you have never worked with these techniques, might be a little bit counterintuitive because you can't see much on the left side while you seem to see here structures like the mitochondria that were labeled here quite well on the right. And if I play the movie, I think it becomes even more apparent, right? So here we see blinking single molecules. I think these were microtubules here. On the right, we have these mitochondria blinking. So the reason why the left data is high quality data is because it's very easy to identify isolated single molecules here, and our algorithms to then find the position of the individual molecules work very reliably here. On the right, the problem is that the molecular density of the active molecules is so high that it's actually difficult to find the molecules you're really interested in. In fact, you actually create a bias that these individual molecules here outside the background are more easily localizable than the ones on your actual target, because these are nice isolated molecules, while here, where the molecules are very dense on the mitochondria, you need to exclude most of them in the localization process. So I think a good rule of thumb is, so if you can recognize the labeled structure already in the raw data while you acquire the data, you should be alarmed, and you should probably consider changing your imaging conditions to get better raw data quality. You can do that by changing the laser intensity, working with the camera frame rate, looking more closely at the buffer you're using, or maybe using a different dye. So after acquiring now this raw data, you need to analyze it, right? This is not the end result we are interested in. We want to know the positions of, let's say, 1 million molecules to really form a super resolution image here. And this is essentially the general process you have, right? You have raw data frames. You need to identify that the computer program needs to identify each of these individual molecules here, cut it out, fit a model function to it to determine the center position of that molecule, and then just plot the center positions that we determined here in our final image. And we do this over about 10,000 to 100,000 camera frames typically. So as you can imagine, this is quite intensive, work intensive for the computer, and especially in the beginning, in the early times, these algorithms could be quite slow, and it took much longer to actually analyze the data than recording the data, and that could create a significant bottleneck. Nowadays, we actually have a large number of different algorithms, and I think this has become better. And as I pointed out in the previous slide, talking about the biases, so in these processes, you need to have a number of filtering steps here, right? For example, you need to filter out any cases where two molecules here are too close together. You can already filter them out maybe early on. You might want to filter out two dim molecules where you actually are concerned whether these even are fluorescent molecules blinking, or if this was some background. And then later on the localized molecule side, if the fit really didn't work, if it didn't converge to any reasonable values, you also want to filter those out. So, and this is where actually then the training, your user input in the data analysis process actually becomes quite critical, and you need to be aware that this is something where you as a user will actually have input into and can fine-tune your data processing. it's this slide is more of a philosophical argument here, right? So you need to keep in mind when you use these super resolution techniques is what's the question I want to answer, and depending on the question, you'll have very different challenges. So I showed you the nuclear complexes at the beginning here as a kind of a simulation or to motivate this. And this is essentially one of the easiest examples. If you want to image nuclear pore complexes, until recently at least, the community was convinced that nuclear pore complexes all look the same. So you can image 100 nuclear pore complexes and can apply, for example, single particle averaging techniques to get the perfect image of a nuclear pore complex. In that case, you don't need to have a particularly high localization density of each nuclear pore complex. It's fine if you only localize 10 of the 32 copies here of a particular nuclear pore in here. And you, in the end, you still get an excellent image here. Another question might be that you look at some membrane patch or a synapse or so, and you want to see clustering of particular proteins. Do they cluster? How big are these clusters? How many copies of that molecule are in the cluster? And here you face challenges, like, for example, to even identify what is a cluster. Everything just is clouds of localization. So what makes this a cluster? Well, but let's me exclude these other individual localizations here, right? Or should that cluster actually include this localization here as well? So you need to work on implementing clustering algorithms then to get reliable data here. And then and this is the ultimate challenge here is, let's say you want to image densely labeled chromatin, and there's still some background here, right? This would be the underlying ground truth, but if this is the data you are getting here, it becomes very hard to kind of make any sense of that. So depending on what you want to image, the difficulty of using these techniques can vary dramatically. So with this, I want to switch over to kind of 3D imaging. So what I've shown you so far was all based on essentially acquiring a 2D image of single molecules here, right? And so this would be the perfect Poinsett function in 2D, and you can imagine this being pixelated, and you find the center position of that. But how do we actually now get the Z information from that molecule? The depth information, right? So what we would traditionally do in microscopy is we take a 3D stack. For the confocal microscope, we do that all the time. But this doesn't work here because the molecules blink. The molecule, if you go from one frame to another through that Z stack, the molecule might have turned on or turned off, right? So you will not get any useful information from that. So you need to be able to extract the single molecule, the 3D position of that molecule from a single snapshot of that molecule. And you actually face two challenges here in that context. And for that, I want to show you a side view of our Poinsett function of the microscope. So if you would be able to look at that molecule from the side, or if you would be able to take a 3D stack and then take a cross-section through that 3D stack, this is how our Poinsett function of that single molecule would look like. And you can see two problems here. First is that the top half looks very similar to the bottom half. So if, let's say, if you take a 2D image at this position, which would say that your molecule is out of focus 300 nanometer below the focal plane, it looks the same as if you take an image down here, which would mean that the molecule is 300 nanometer above the plane. So you have that symmetry problem of the Poinsett function. And the second problem is that if you stay close to that focal center here, this is about a 700 nanometer or so distance here. Within the inner 300 nanometer or so, the Poinsett function hardly changes. And we know that actually all the time when we focus our samples on the microscope, let's say beads for calibration purposes or so, it's very hard to kind of do the last little adjustments of that focal plane to get them perfectly in focus. Or in other terms, it doesn't matter too much if it's 100 nanometer above or below, it looks the same. But here, of course, that's exactly what we want to find out. We want to know if a molecule is 100 nanometer higher or 100 nanometer lower because we want a 3D super resolution stack. So we need to solve these two challenges here, right? So the axial symmetry of the Poinsett function and the fact that the Poinsett function doesn't change much in the vicinity of the focal plane. there have been many strategies over the years how to solve this. We are not going into depth here, but this is from an older review, which is about 10 years ago. And since then, there have been quite a number of additional innovative approaches, in particular here by Sandrine Levesque-Ford's lab here, which are not included here in this figure yet. So the trick is that you somehow need to break that symmetry of that Poinsett function, and that you somehow need to create sensitivity in that plane where the molecules are in focus. So if we do a poll essentially just by asking what type of methods are mostly used in the field, we have to, I think, admit that astigmatism is by far the winner at the moment. And the reason for that is it's probably easiest to implement. We contributed to this biplane approach, and I think that's in the Bruker system. That's also here at the meeting. And then the double helix approach and is another very popular approach that particularly was popularized by the Mörner lab. And I'll talk a little bit more about these 4-pi interference approaches. And then there are other techniques, and some might actually overcome here the dominance by astigmatism in the next years. Who knows? But just briefly, since astigmatism is so popular, I just want to briefly show you how that works. It's also easiest to explain in a way. So you have your objective and your tube lens, right? And that creates that nice image of a single molecule. And now you distort it by adding a cylindrical lens in that beam path. The cylindrical lens will add astigmatic aberrations into your detection beam path. And what that means is that the light being in the plane here parallel to that screen here will be focused a little bit more strongly than the light in the light rays here in the plane perpendicular to the screen here. So, and that leads to the fact that if you go a little bit above or below the focus, the Poinsett function will be focused more in one direction than the other. So here you see this elliptical pattern in a vertical orientation, and here you see it in the horizontal orientation. So what you now can do is you analyze the width of that Poinsett function in the x and the y direction separately, and you see that you have kind of unique ratios of these two widths here, which then correspond to a particular z position. So that's how you extract then the z position of that molecule. And this is here a raw data set, just to give you an idea how the raw data with astigmatic imaging looks like. The effect can be quite subtle, but the computer algorithm will still be able to identify it relatively reliably. So let's see if I can pause this. I think you can see in this case most of the molecules are extended a little bit in the z direction here. So probably the sample was probably out of the focal plane a little bit in general here. So the resolution that we get with these, with most of these approaches in the z direction is about 2 to 3 times or 2.5 to 4 times or so worse than the resolution we get in the xy direction. So why is that the case? And the reason lies in that Poinsett function that we looked at earlier, and the formula I showed you at the very beginning. The localization precision is proportional to the size of the Poinsett function, and the Poinsett function is longer in the z direction than it is wide in the focal plane. And that same proportionality is then reflecting in these localization precisions or the resolution of single molecule localization microscopy. Why is the Poinsett function elongated? It is elongated because we only have a single objective that only collects light essentially from this one angle, this solid angle here defined by the numerical aperture here. So the idea then is, would that improve if we add a second objective lens here? So now we can actually get an interference effect here between the two objectives, and we get a Poinsett function which is known from the confocal 4-pi microscope and the I5M developments in the 1990s, which looks like this. And now this is much sharper here in this axial direction than it was before, right? So this is the early 4-pi papers here, which Kate mentioned I worked on during my PhD. And then this was then realized that this can be also used for single molecule localization microscopy first here in the IPALM paper by Harald Hesse's lab, and then Stefan Hell's lab picked this up as well around the same time with this 4-pi SMS microscope. And we then built a kind of a version 2 essentially of this 4-pi SMS microscope. I want to show you some data of. So the Poincare function gets about 5 to 7 times or so sharper here in the axial direction, and that results then in a better Z localization precision in particular, and we get roughly 20 nanometers or so in all 3 directions, maybe a little bit better. So how does this microscope work? How does this look like? Okay. So you have these 2 objective lenses, and you have a single molecule here in the middle. The fluorescence emitted by the molecule is collected by both objectives, and then it's recombined here at this beam splitter cube. And this beam path and this beam path have been carefully adjusted to be of essentially the same length here. So the light now can interfere here that was emitted by the single molecule here. As a physicist, I'm always really excited about this because we are really seeing here the interference of single photons getting emitted here from that one molecule, because fluorescence itself is incoherent. So it must be that a single photon interference that we see here, and then added up over many, many photons, we get that interference effect. So if this beam path here and this beam path here is exactly equal of length, then we get constructive interference, and we get a bright signal coming out of this beam splitter cube side. That also means that there's no energy left to come out on this side, and we get zero interference or destructive interference out on this side. Now, if we can look at different polarization directions, and there's a little trick we can play by putting a birefringent quartz wedge in here, and that means we can shift one polarization direction, the interference, a little bit versus the other. So this would be now positive, perfect constructive interference, 0-degree phase shift. This is the 180-degree phase shift, and here would be 90 degrees and 270-degree phase shift. So we get these 4 images out fairly easily now with this beam splitter cube, and then this birefringent element. So what happens now if the molecule moves a little bit up and down between the 2 objectives? If it moves up and down, you see that the interference patterns will change, right? Because now this beam path gets a little bit shorter, this one gets a little bit longer, and we get these the sinusoidal behavior of these 4 intensities here, depending on the position of the molecule. And extracting now the intensity ratios between these 4 allows us to determine the axial position here. So in practice, this setup looks more like this here. This is the 2 objectives. This is beam splitter cube. Here's this birefringent element, or here. And then this part is just an effort to recombine these 4 images we have next to each other onto a single camera chip, so that we don't need to buy 4 cameras. And then this is how the setup in our lab then looks like in practice here. This is the 3D SolidWorks design. Most of the interesting part happens here in the center around the objective lens, where you need to move objectives and stages very precisely relative to each other. So yeah, what we added at the time was we added some adaptive optics with deformed mirrors to allow imaging of thicker samples here over the earlier approaches. And this is an animation of a synaptic complex data set of a mouse spermatocyte. And I think this also shows nicely how we acquire a sample which is about 10 microns thick. So you see here, I mean, this is simplified in the animation, but we see the molecules blinking, right? We can localize them over a range of roughly a micrometer. But then we can, of course, move the Z stage every 1,000 or 10,000 frames and image molecules a little bit deeper in the sample. And then, as we do with a confocal microscope, acquire a 3D data set by essentially just stitching all these together here. So that gives us then a data set like this. Where we can now localize here the elements of the lateral elements here of the synaptic complex here, or SYCP3, I think it was here, quite precisely with get about 20 nanometer resolution, easily sufficient to resolve that distance of 100 to 200 nanometers between the two lateral elements, no matter of which orientation the synaptic complex or the has here in this particular sample. And then you can do some funny, well, I think this is quite fitting here to the our view here towards the ocean with all the ramps and slides here. So this is quite old data, but we have this microscope now for nearly 10 years in the lab, and it's still working strongly, which I'm really proud of. I can say that of all our microscopes. So this is a really beautiful application that Maohan Su, a postdoc, shared between the Rothman lab at Yale and in my lab, has worked on now over the last few years. And this is about the compartmental organization of the Golgi apparatus. This is a focus of the Rothman lab for decades, a focus of our department of cell biology at Yale, which focuses a lot on the secretory pathway as well. So this is really at the heart of what our community does here. And what is known so far is that key components in organizing the stacking of these Golgi cisterna are these Golgi matrix proteins. And this is from a review paper from Sean Munro's lab. And you can see that there are quite a number of them, but very little is known about them, right? So we know they're long coiled proteins. They're very flexible, but we don't know the crystal structure. They're also densely localized around the Golgi, and there has been some evidence, but not necessarily concluding evidence, that they are localized. The different Golgins are localized to different cisterna of the Golgi. We know also that they are heavily involved in tethering of vesicles and then directing or moving them towards the Golgi so that they can fuse with the individual cisterna. So the problem why we don't know much more is that the Golgi cisterna are very close together. They're about 65 nanometers separated from each other. So it's really difficult to resolve that with a light microscope, even with a 2D super resolution microscope, because the Golgi is, as you'll see in the next few slides, often is this really convoluted ribbon that is folded and twisted in 3D around the nucleus. So we applied the 4Pi SMS microscope in our lab for that. And Maohan has worked on this for a couple of years, actually, to get this to work. So here's a typical data set that Maohan acquired. And this shows how the Golgi looks like in a typical cell here, like for example, a HeLa cell. You see here two different Golgins. This is GM130, and this is a sheet Golgin, which is on the cis side of the Golgi stack. And then giantin, which obviously forms a ring around rim, is on the rim of the Golgi cisterna. And I think if you look at this in 3D, I think it becomes quite apparent how twisted this is. What you can see here is that these look very flat here, because we are not don't label the whole Golgi stack. These, as it turns out, only label the first cisterna of the Golgi. That's why it looks like a very thin ribbon here. So what we can then do is take cross sections here of these individual cisterna and measure either in a cross section here along the sheet or across kind of the stack of the sheets, right? We call that here in the side view here. And if you then look closely, it actually turns out that GM130 and giantin are not exactly in the same plane. So there's a little shift between these two. And this, of course, was corrected for any chromatic aberrations here. So Maohan then actually systematically did this for a large number of, essentially nearly all Golgins here. And you can see here already in this overview that the distance, he always used the GM130 as the reference, and then you can see here, sorted this way, that the distance gets bigger and bigger here. The GCC 88, for example, there's a clear separation between the GM130 and here this layer. And you can quantify this over many cells, measure the distance, but what's also apparent is the width of these also varies here. And Mauan summarized this here in these two figures here, which has tons and tons of data that went into this. So this is always the distance to the GM130, which is the cis phase of the Golgi. And you see that GMAP 210, for example, is in the same layer. Golgi 84 is about 60 nanometers separated. Then there's a 120 nanometer separation. Then the next two are somewhere in between. And then there's a bigger jump to about 180 nanometers. And when we measure the width of these, you actually now see why these are in between here. These are somewhere in between the first and the 1 or 2 cisterna, and the reason is because they're thicker. They are 2 cisterna thick. So they are actually, so these giantins are localized in 2 cisterna. So I'm cutting this short here, since this is primarily about the microscopy. This is in a bioRxiv paper published a couple of months ago. We are currently working on the revisions, but I'll just want to end with kind of the model that Mauan created from this. So using this 4Pi 3D super resolution imaging, we were able to really map now all these Golgians across the Golgi. It's clear that there are 4 layers, not 3, not 5, and that they are clearly arranged in these individual cisterna. Some of them are localized in 2 cisterna, or on 2 layers, while others are clearly defined to just one of them. And Mauan has now validated this further by knocking out a few of them. And for example, you get rid of 1 layer, etc., etc. So what I didn't talk about so far is multicolor imaging, right? So obviously the data you've seen on the last few slides was already 2-color data here. And the classical way how this, you could imagine doing this is how we would normally do it on any confocal microscope, is you take a far red dye, you take an orange or red dye, right, and you image them in different color channels. This has a couple of issues though, and there are better approaches, I think, in the single molecule localization field. But this is the traditional approach, right? You take Alexa 647, you take Sci3B, you get 2 images. This is from that initial paper I talked about. But this looks good if you look from afar, but the closer you look, it's not that impressive actually. And the reason for that is that the good dyes, the ones that blink really well in the storm approach, they're all in the same color range. They're all in the 647-ish Sci5 Alexa 647 color range. If you go to Sci3B, it also blinks, but it's twice dimmer. It doesn't blink as well. The second problem is you need to image them separately, sequentially, one after the other, because otherwise one laser would activate the other one, and the blinking would be messed up. And then that creates drift issues. And then you have chromatic aberrations. Different colors will be slightly shifted, maybe by 50 nanometers, maybe by 100 nanometers, but that's obviously a resolution range that we are really interested in. The solution is ratiometric multicolor single molecule localization microscopy here, and this is this is what Mauan actually used in the data that I just showed you, and this is quite nicely summarized here by this paper from the EVAS lab here. So, for example, you can image Alexa 647 and the CF680 dye, which by essentially just putting a dichroic beam splitter in between and then imaging the 2 color channels at the same time on a camera chip. And here you see the two images acquired with the camera, and you can see that a few molecules appear brighter in one channel than in the other one, right? And now if you plot the ratio between the brightnesses, you see these 2 peaks. So if you are an experienced multicolor diffraction limited microscopist, you would say 647 and 680, that's a challenge, right? Normally you would not choose that. They're a little bit too close together. You can use linear unmixing techniques, but they will not be great. But this is much easier here, and that's the good news, because you don't try to do a kind of an analog unmixing where you try to determine what ratio of the dye in this pixel, what ratio is in this channel, what ratio is in the other channel. You have to make just a binary decision, because you see single molecules, and you know it's either one molecule or the other molecule. It cannot be a mix of the two. And that makes that decision easier, and that's why we can relax these conditions much more. And even if the dyes are only 30 nanometers or so separated in spectrum, you can easily distinguish them. I mean, in our case, we can do 3-color imaging and still only throw away, so using 3 dyes essentially in this color spectrum, still only throw away about 5% of the localizations, which are just here at this minimum there where it's ambiguous which dye they are. Another approach to do multicolor imaging is using the DNA paint approach. And I have not introduced that so far here, so I could have introduced that earlier, but I think it makes maybe a little more sense here now. So DNA paint is this other way of doing single molecule localization microscopy. And here we use binding and unbinding of single-stranded DNA probes, binding to the complementary sequence which is bound to the target of interest via an antibody typically. So you have what we call an imager probe here with a dye attached to it, and that diffuses around very quickly. It's a small molecule, and it diffuses so quickly that we can't see it on our camera chip. It essentially just creates a little bit of blurry background. When it binds, however, it will get immobile, and during that time, we see it appear as a flash of light that we can detect, similar in the same way we got the Palm Storm, D-Storm images before. We call that the docking strand here it attaches to. So we get the same switching essentially now through that association and disassociation of the imager probes. The advantage over the Palm and Storm approaches, and that's why I'm a big fan of DNA paint, is it's easily engineerable. The blinking properties, so the brightness of the dye or the brightness of the probe is only depending on the dye. The blinking properties depend on the DNA, so you can modify one or the other, right? So if you say, oh, they bind a little bit too long, well, then you just remove a base pair. If it's too short, you add a base pair, right? So you can also easily design orthogonal base pairs so that you can do multicolor imaging, as I'll show in a minute. It's also resistant to photo bleaching, because if one of these dyes bleaches, the DNA will fall off anyway, and you can replenish it through by new imager probes from the solution. In the past, this DNA paint, and that's I think why it took a while to become popular, is it was not very fast. The blinking was very slow. People imaged at 4 frames a second instead of like 100 frames a second or so, what we do in Storm, and it took hours to get a good image. And the reason for that was the background was too high. A single molecule did not create a lot of background, but if many imager probes diffusing around in solution, that creates a substantial background. So you had to keep that low enough, or you had to go to turf imaging, but that limits yourself to just the cover slip surface. So with the advent of fluorogenic imager probes like these ones that we published a couple of years ago, or probes which have a very high association rate, where you can then keep the imager concentration low, like the ones that Ralf Jungmann has published, and he'll give a talk, I guess, tomorrow or in 2 days. This problem has been overcome now, and I think DNA paint is now as fast as the Palm Storm approaches. So now, how can we do multicolor with DNA paint? It's quite straightforward. It's called exchange paint, right? So you just have orthogonal single-stranded DNA docking strands here. So you come in with an imager probe. It will transiently bind to one of the docking strands, but not the other one, because that's not a complementary sequence. And when you're done imaging that, you wash it out, and you come in with the next imager probe, and that will now bind to the next target, and then you can do that to a third, a fourth, a fifth, 10th target. So this is a great concept. It works really well. The only problem is if you have really highly multiplexed imaging approaches. We are currently aiming, can we do 100 different targets or so? So the problem there is it scales linearly in cost, right? So this says $400 per probe. Now we're actually paying $800 per probe. So if you want 100 of them, you would need to pay $80,000. It requires this washing between imager probes, imaging rounds, and that of course will damage the sample if you do that 100 times. And then the real killer is, however, and I don't have time, unfortunately, to go into this, is the sequence space is relatively limited with these new generation of fluorogenic or speed imager probes that I briefly mentioned, which are the solution for modern DNA paint imaging. We have reduced the possibility of sequences to somewhere between 5 and 10 actually now. So we cannot easily scale this up to 100. So what Florian Schüler in my lab, a postdoc at the time, who actually came from Ralph's lab where he did his PhD, and now he's setting up his lab at EPFL in Lausanne, is developed this flash paint concept. And what we did here is we added what we call a transient adapter in between the docking strand and the imager probe. So the transient adapter will bind with 1/2 to the docking strand, and the imager probe can now bind to the other end of that transient adapter. So both of these interactions are transient, and this seems like a big disadvantage because now you need two things to coincide to get a single blinking event. But the good thing is you can tune this to essentially not be a problem. The transient adapters bind for tens of seconds, 10 to 100 seconds, and can be used at very high concentration because they are not fluorescent by themselves. So 80% of the docking strands are occupied by a transient adapter at any time, while the imager probe binds for 1/10 of a second, so gets this fast blinking effect by just the imager probe to the adapter binding here. So when you're now done imaging that first target, you don't need to actually wash out that cocktail. You add what we call an eraser against the first transient adapter, and at the same time a new transient adapter. So the eraser will bind to the earlier transient adapter, and since the transient adapter is binding transiently to the target, you just need to wait for 100 seconds or so. It will have come off anyway of the target. The eraser is in high abundance. It will capture all the adapter and essentially neutralizes it. It also blocks the imager from binding to it. And then the same imager will now be directed to the new transient adapter and bind to the new target that way. So without washing, you can now redirect that imager probe from one to the other. So now you need to buy 100 adapters, and 100 erasers, but can use one imager for 100 color imaging, and the adapters are cheap, right? There's no particle modification. It's the same things we buy as oligos every day for our molecular biology experiments. There's no washing required, no bleaching required, and the erasing works remarkably efficiently. And here, let me show you this in a just a spinning disk microscope setting. So we switch here from microtubules, from mitochondrial labeling to microtubule labeling by just adding that eraser and second adapter at the beginning of that movie. Let me replay this. So here you just see the mitochondria. This is highly I increase the contrast here to show that there's not much background. And then within a few tens of seconds or a couple of minutes, it switches. This is a 40-fold sped up. And so you could not really see the microtubules in the beginning. Now the mitochondria have completely disappeared. So this works extremely efficiently. And I'm speeding up a little bit because I'm out of time here. But we have 12 color imaging here of the Golgi complex, which I already introduced you to here. And so we can now see here within a single cell, we can see the stacking here of the Golgi with the cis medial trans-Golgi cisterna and then the trans-Golgi network. We can move to a different area where we now see COP2 vesicles and tubules budding off the exit sites, which are marked by Tango 1. COP1 vesicles are different locations here surrounding the Golgi. And I already introduced you to these rim Golgians, right? So which can nicely be shown in the same data set. And then, of course, you can look at lamin and other labels at the same time. So in summary, let me just summarize this and contrast this maybe to other super resolution approaches here. The strength of single molecule localization microscopy I would argue it still is the technique with the best 3D resolution in particular. STED and single structure elimination microscopy will normally not achieve these 3D resolution levels. We'll hear from Francisco in a second about MinFlux, which is also a single molecule localization microscopy technique, I would argue, where the resolution gets down to sub-nanometer precision. So there's really no end to the resolution you can get with these techniques. It is a single molecule technique, so you can technically count single molecules. I didn't go into it. It's not as easy as it sounds, but it's doable. And then and the multiplexing approaches with the DNA paint are really quite promising. The difficulties are that you really need to carefully select the fluorescent probes and often the imaging buffer. Do not assume you can use your standard confocal microscope slide, just put it on there and it will work. It might work for structural elimination microscopy and area disk, area scan microscopy, but not here. It's also not super fast. So if people ask, can I do fast live cell imaging? It comes at severe compromises. I would say a typical recording time is in the order of 5 minutes to an hour. So you need a stationary sample. And the training on the image processing side should not be neglected. You can easily create artifacts of localizing the wrong molecules if you don't know what you're doing here. But with that, I'm at the end, over my time, and happy to take questions if there's time.
So thank you so much for the lovely talk and the very lovely images. I wanted to start with a quick kind of basic question. When we compare 2D storm imaging with your 4-pi technique, for example, 2D storm, we kind of expect at least 10,000 images per acquisition. How does that change for your 3D 4-pi configuration?
It doesn't. So we just have 4 images taken at the same time on the same camera chip, but we take still 10,000 or more realistically probably 100,000 images if you want a high quality 3D data set. Yeah.
So the acquisition time is similar?
So it would take, I would say it's typically in the tens of minutes to an hour range. Yeah.
Thank you. Are there any other questions?
Hi, thank you. I have a question about your resolution. You mentioned here, you said 25 nanometer resolution. And are you trying to reduce your targeting objects because you use antibody, nanobody, then DNA strain, which I think in total would be like 15 or 20 nanometer. So basically your 304 is quite far from your target.
You're right.
Are you trying to optimize this or to reduce all those objects?
You're absolutely correct. At this resolution scale, and I think Francisco will probably talk more about it because that's really important for MinFlux, you need to consider the size of your probes, right? So they become at the size scale of the resolution. So for many probes, fortunately, it doesn't matter too much because there's a certain level of flexibility and over time, or like over a second or so, where you localize the molecule, it will have somewhat averaged out. It will not be perfect, but it works reasonably well. But you always need to keep that in the back of your mind before you really draw conclusions where you say, well, these are 12 nanometers apart. It might be a little bit more, a little bit less. So yeah, you need to be careful there. Nanobodies help, right? Using smaller probes there. The DNA paint probes, we often put the dye at the end facing the nanobody rather than the other side. But in practice, however, it's the rotational flexibility or so is fortunately averaging out many of these problems.
Thank you.
And we might have time for one more question.
I have a question, maybe a bit naive, about DNA paint compared to fluorescence in situ hybridization. And why would you use one compared to the other? And a bit linked, can you do RNA paint?
You're absolutely right. So fish and DNA paint uses the same interaction of 2 single-stranded DNA interacting with each other. The difference essentially is just the number of base pairs. So in fish, I think you typically have 30 or so base pairs, while we have 6 to 10. And each base pair adds a factor of 10 roughly as a rule of thumb in binding affinity, or like it binds 10 times longer. So this is now 20 orders of magnitude longer bound if you have a fish probe versus here, which means eternity versus less than a second. And that's the difference, right? So we want the transient binding. In fish, you don't want transient binding. You want this to stick forever. But it also means, and then of course, if it's DNA fish, you first need to melt your DNAs to be actually get access to a single-strand DNA. Could you directly bind this to RNA? I think yes. Yeah, if you, I'm blanking right now if this has already been done, but yeah. Okay. Well, Francisco will show it. Yes. Yeah.
Well, thank you again for the nice talk.
Okay.
In Argentina before doing a PhD at the University of Buenos Aires, focusing on photonics, optical nanolithography, superlenses, and plasmonics. Also, coincidentally, also did a postdoc at the Max Planck Institute of Biophysical Chemistry, and is currently a group leader at the Research Institute for Molecular Pathology in Austria. And he has several interests, including interdisciplinary approaches to try to solve some of these regular problems or issues when we look at biological samples, and particularly has interest in super resolution techniques such as minflux and nanoscopy.
Sorry. Very sorry.
We also have an interest in solving IT problems.
You go for it. So meanwhile, I want to thank the organizers for inviting me and including this topic in the program. This place is amazing. I want to thank Joerg for introducing many of the content, so I can do less work on that side. Let's see. We have a challenging situation with, I'm not present with my computer. We have lunch coming up and your caffeine has decreased. So let's see how well we can do this. So I will just start anyway, well, or not. No, I don't start. Can tell you some interesting stories about something.